My name is Luis Saman, and I'm a PhD student working with Charles Ofria and Rich Lenski. So Charles is a computer scientist and Rich is a microbiologist. And my work actually combines the two fairly well. I'm interested in post-parasite coevolution, and with Charles I usually work on a system called AVITA, which is a way of studying evolution using self-replicating computer programs. And I'm interested in host-parasite coevolution, so I use both host and parasite self-replicating computer programs. Parasites infect the host, steal their CPU cycles in order to reproduce, and using this we can ask questions about fundamental processes in coevolution. One of those questions involves sort of how does coevolution produce the complex community of interacting hosts and parasites that we often see in nature. With my wet lab work, I'm working in Rich Lenski's lab, who is well known for his long-term evolution experiment where 12 replicate populations of E. coli have been evolving in the lab for 58,000 generations. The experiment started in 1988, I believe, so it's a long evolution experiment, one of the longest. And in the lab I'm also working on host-parasite coevolution. In this case I'm using viruses that infect bacteria.
My name is Madeline. I'm an undergrad at the University of Texas. I've been working this summer at MSU with Dave Bryson and Aaron Wagner. So I got to do some software development to help us analyze some of our results and learn more about what we saw, and then built a robot using Lego Mindstorms to replicate the behavior we saw in a digital organism with an actual robot. So Beacon has given me the opportunity to come to a different state and work on a really cool project.
Hello, my name is Kiana Weatherspoon. I'm a senior here at MSU, and my major is physiology.
Hello, my name is Maya Rolls. I am a human biology here at Michigan State University, and I am a senior.
And we both work in Dr. Richard Lenski's lab, and what our research is based off of is Dr. Richard Lenski's long-term evolution experiment, and we work under Dr. Zachary Blount, and we specifically study speciation, well, possible speciation, in one of the populations from the long-term evolutionary line.
Hi, my name is Wesley Lofty-Eaton. I'm a postdoc working together with Eva Top at the University of Otto in Moscow, Otto. And our approach to studying evolution in action is looking at plasmids and the general mechanisms or strategies employed by plasmids in order to adapt to new bacterial hosts under selective conditions.
Hello, my name is Jared Moore. I'm a second-year PhD student in Dr. McKinley's lab, and I work with evolutionary robotics. So what we try to do is work with simulation and actual hardware prototypes, and use evolutionary approaches to develop robotics for aquatic sensing. What I got in front of me here, a couple of our 3D printed prototypes using Arduino hardware with Evolve controllers on them that we plan to use in our testing facility to test out solutions we develop in simulation.
I'm Aaron Wagner. I'm a postdoc in Charles Ofria's lab at MSU. I joined Beacon when it first started. I used to be a field ecologist studying the evolution of group formation in striped hyenas in Kenya. I came to work in Charles's lab because I wanted to be able to follow up on some of the field research that we couldn't do because obviously a striped hyena system is a very slowly evolving system.
I'm Brian Connolly. I'm a postdoc in Ben Kerr's lab at the University of Washington. I do research on the evolution of cooperation and the coevolution of cooperation with communication. I try to do that in multiple systems. So I'm originally a computer scientist, and through collaborations that I met with Beacon, I started doing wet-bench work with microorganisms, and that's kind of led me in a whole different career path. So it's really been nice to be able to combine both approaches and really get a bigger picture view of the kind of questions I'm interested in.